Welcome to the STEMIT Learning Program. STEMIT is a program that teaches mathematics, physics, chemistry, and biology. In short, STEM abbreviates science, technology, engineering, and mathematics. This program is available both online and on television. We offer the following: electronic books, PhET simulations, syllabus documents, and special educators. Now, let's see how this program runs with these simulations.
Greetings everybody. I hope you're all keeping well and keeping safe in these times of the COVID-19. We are today going to look at the enzymes as a topic in the LGCSE syllabus. With emphasis, we are going to be looking at enzymes as catalyzers of reactions. We are going to be looking at enzymes in terms of trying to define them, understand the structure of enzymes, and we're going to look into what can affect the activity of enzymes, so on and so forth, until we can go into checking the uses of enzymes in industry, how they are being used, so that you learn and understand why you should learn about enzymes, and you should learn into the practical world as to where enzymes are used. And if we may continue into that, we can define an enzyme basically as a protein body. This protein body, as we're looking on our screen, we can say this as an enzyme. It has got what is labeled as an active site. And if you look closely, an active site has got a peculiar shape in which it has. Whatever that the enzyme is going to be working on is called a substrate. Now, the enzyme is a protein body that speeds up chemical reactions. So the raw materials in a chemical reactions are called the substrate when we are emphasizing the issue of enzymes. Now, an enzyme will have an active site that has a perfectly integrating shape towards its substrate. Now, if we'll be having an active site like that on our enzyme, and then the substrate comes and binds into our enzyme, we're going to have an enzyme substrate complex as illustrated here. Now, take note, the shape of our enzyme, which does not have to change, it doesn't change, will attract the substrate, and enzyme changes shape slightly as substrate binds to it, so that it merges perfectly into it, to facilitate the merging of the enzyme into it. And then as you move further, the enzyme product complex, what does it say? It says now our substrate has been broken into two. This is why now it is a product. It is no longer a raw material. And if you look again as it proceeds, the chemical reaction proceeds, you'll find that products leaving active site of an enzyme. Now let's look at this. We said this is the active site of our enzyme. It is still looking perfectly as it was in the first stage before it binded to the substrate, creating an enzyme substrate complex. It is said that when it got here to after binding to the substrate, the enzyme shape will change slightly. The slight change is just to accommodate and put in a fixed position the substrate. After the product has now been procured, we have been attained from the substrate, the enzyme is still maintaining the very old shape, previous shape that it started with. So basically, what are we saying? The diagram here should tell us that an enzyme will actually have an active site that has a special shape to the type of substrate or raw material in which it facilitates its reaction. Because the enzyme is not part of a reaction, but it only makes a reaction to occur faster. In technical terms, we say the enzyme lowers the activation energy that is required for a reaction to occur, but it is not part of the reaction. We must note that a reaction would still occur even without the presence of an enzyme, but the enzyme only speeds up the reaction. A better word for the speed up is the enzyme catalyzes the reaction. Therefore, the enzyme acts as a catalyst to the chemical reaction. Therefore, to put in mind is it speeds up the reaction, meaning it catalyzes the reaction. Therefore, it is a catalyst, and it is not involved in the reaction. It binds to the substrate, and when the reaction is done, it is free to go and bind to other substrate molecules. It does not get involved in the reaction. It does not get consumed in the reaction when it catalyzes. So, since we said this is a protein body, the enzyme, this protein body enzymes, they could be affected by several things, and to highlight, they could be affected by temperature, they could be affected by pH, acidity or alkalinity, in whatever solution in which they are working. Now, let us try to explain this concept. Now, we see that the enzyme has got a special shape, which is very specific to the substrate that it will catalyze its reaction, that it will bind to. The technical phrase that is used to describe this perfectly integrating shape of a substrate and an enzyme, it is the lock and key hypothesis. If we were to explain this, we say a particular key will lock and unlock its specific lock. It is not any other key that you can just take and unlock whatever lock, but it is its special key that will unlock it because of the shapes, which in this case we know that because of the peculiar shapes of the active site of an enzyme, it will be able to bind to a certain substrate that has got a peculiar shape that will make it bind to it. So, what are we saying? If now we have to proceed to the issues of temperature and pH, we say, let us say given a higher temperature or lower temperature than what the enzyme prefers, and by the way, the temperature in which an enzyme works best is called an optimal temperature. So as to say, even the pH in which an enzyme will work best is called an optimal pH. Now, if the temperature, let us say it is way beyond the optimal temperature of the enzyme or way below the optimal temperature, you will find that the active site in which the substrate is supposed to bind will be distorted. It will change that shape because of that temperature that is beyond the optimal temperature. The change of the shape of this active site, the terminology that is used there is said that it is denatured, meaning the shape here is now altered. It is denatured. Therefore, temperature that is outside the optimal range temperature for an enzyme to work will denature the active site of the enzyme. Therefore, when the site is denatured, meaning the shape is distorted, it will no longer be able to bind to that specific substrate to create the enzyme substrate complex. Therefore, it will not be able to speed up the reaction anymore. It will not be able to catalyze the reaction anymore when the shape of the active site is changed. Let us also explain the issue of pH, level of acidity of alkalinity in which an enzyme will be catalyzing reactions. We know that they are made of proteins. Proteins are made of amino acids. Now, let us say in case the there is a lot of acidity in the solution in which the enzyme is working. The hydrogen ions that will be there because of increased acidity will cause reaction to the amino acids that make up the protein of the enzyme. The moment the amino acids are ionized, they will change form, which will eventually change the shape of the active site. And the moment the shape of the active site changes, it will no longer be able to bind to the specific substrate in which it was meant to catalyze its reaction. Therefore, whenever that happens, you will not be able to achieve the enzyme substrate complex, which then will say your reaction will no longer be catalyzed or sped up as the enzyme is supposed to. So basically that is how we explain the lock and key hypothesis, saying it is by virtue of the active site of an enzyme having a peculiar shape that will bind to a specific substrate to form an enzyme substrate complex. And the moment that shape of the active site can be changed, and what can change it will be temperature that may be higher or lower the optimal temperature of that enzyme, or pH that may be higher or lower the optimal pH of that enzyme. And the moment the shape of the active site changes, we say that enzyme has been denatured. And what is denatured, it will not be able to bind to the substrate. And if it cannot bind to the substrate, we cannot have the enzyme substrate complex. Therefore, the enzyme will not be able to catalyze that reaction anymore. Since we have discussed the structure of the enzymes, how they work, how they could be nurtured, having that understanding, it would be beneficial for us to discuss a little bit on the uses of the enzymes. The first use of the enzymes that we are going to be talking about is an example of an enzyme called pectinase. As an enzyme, pectinase acts on pectin. Pectin is a substance that it is found in most fruits. It has the properties, if it is heated together with sugars, it forms a gelatinous substances. Pectin is used in the jellies that we eat and the likes. So now, enzymes, pectin is acted on an enzyme called pectinase, and this pectinase acts on pectin, breaking it down. When in industry, things like production of wine, if we had pectin still present in the wines, we would have the wines very cloudy and milky kind of. But the enzyme pectinase breaks down the pectin. This is why we have wines as perfectly clear as they are. Even in the extraction of juices from fruits, there is pectinase acted, and pectinase is added into the solutions in which the extraction occurs, and we have clearer juices that are free of pectin. This is one example whereby enzymes are used. A very particular instance that we're going to look at right now is an issue of penicillin. Penicillin is an antibiotic. What are antibiotics used for? For example, in long time back, there was a scientist called Alexander Fleming. They discovered that wounds delay to heal in many years back because of the activity of bacteria. Now, what they discovered was that a group of fungi called the Penicillium notatum, which is a fungi, produced a substance that is called penicillin. Now, this penicillin aided towards the quick and faster healing of wounds because it did not encourage the production of bacteria that infected the wounds. Now, this time around, we are looking at enzymes, but we are going to look at a scenario whereby enzymes are inhibited to work. Enzymes are inhibited to work, but as a function that we need. Now, penicillin is an antibacterial. Why is it an antibacterial? On our screen right here, we are going to look at something. Here is a structure that is drawn in red. This is an enzyme. Now, this enzyme has got this active sites in which it is supposed to bind these two molecules as substrates. Now, the reaction is supposed to proceed whereby we are going to have this having acted on and having catalyzed by an enzyme. Now, the problem right here is in this active sites in which the enzyme is supposed to bind to the substrate and have an enzyme substrate complex, we have penicillin filling into the active site. Now, penicillin is inhibiting this enzyme to catalyze this reaction. This reaction is a reaction that is supposed to assist the production of bacteria in building their cell wall. Now, with the presence of this penicillin, and then it inhibits the binding of the substrate towards the active site of the enzyme, we do not have a cell wall being produced. Therefore, this inhibits the production or the growth of bacteria, multiplication of bacteria by inhibiting production of the cell wall. When bacteria is built without a proper cell wall, it will suffer from osmosis processes, and it will eventually lose out of water because of not having a proper cell wall. Therefore, the growth of and multiplication of bacteria, let us say for instance in a wound, will be inhibited, and the wound will heal much faster without bacteria because penicillin managed to inhibit the active site of the enzyme, therefore not allowing the production of cell wall for the bacteria to multiply. This is where one instance whereby we have seen the enzymes catalyzing reactions, but now we have a reaction of bacterial reproduction that is catalyzed by an enzyme, and then we have this penicillin that inhibits that particular process whereby the cell wall of bacteria will be made. This is one example whereby enzymes are involved, and we should pay thanks to the scientist Alexander Fleming, because as it was discovered that penicillin inhibits the production of cell wall. The first discovery was that it was found that whereby in an agar of growing bacteria, whereby fungi had grown, there was no bacteria observed to have grown and multiplied there. Meaning the fungi had a certain substance that it produces as its metabolite that inhibits the multiplication of or reproduction of bacteria. Therefore, it was extracted, this penicillin, from this fungi, the penicillium fungi. The penicillium was extracted, and it is used now. It is sold in the pharmacist in the hospital as a remedy, as an antibiotic. It inhibits the multiplication of bacteria in our bodies, and we have understood how, or we have discussed how it inhibits the production of this bacteria by inhibiting the production of their cell walls, by inhibiting or occupying the active sites on the enzyme that is supposed to catalyze the reaction that produces the cell wall for the bacteria. This is how it is actually works. Now, the production of this penicillin, given the high demand, is in industry produced at a very high scale, but the basic concept is that the penicillin will inhibit and occupy the active site of the enzyme, thereby not promoting the production of cell wall for the bacteria, and the bacteria will not multiply thereof. I think that should settle it for us, folks.
Thank you for watching today, and please contact us on the information below.